In this 5 mark subjective question, they are asking us to give the scientific name of the organism used in Thomas and Morgan experiment. We need to give the correlation information about allele with specific reference to linkage and recombination. For this, first we are mentioning the scientific name of the organism, Drosophila melanogaster. For this, you will be getting 1 mark. And for mentioning the alleles behavior sense in terms of segregation, it's not happening properly because of the alleles existence in the X chromosome. What do you mean about that? The somatic chromosome controlling alleles are there in the X chromosome. That is the reason why it is not showing similarity with the Gregor-John-Mendel's dihybrid ratio. For this mention, you will be getting half mark. And we need to mention tightly related alleles tend to bring the same parental character in most of the instance, whereas in few instances it is had the probability of getting recombination inheritance as well, but that probability is very less. For this mention, you will be getting half mark. For mentioning the loosely linked allele, bringing the recombination or new combination instead of bringing the parental combination, for this mention you will be getting half mark. And for mentioning the gene present on the same chromosome are said to be linked, and the recombination frequency depends upon the relative distance on the chromosome. For this mention you will be getting another half mark. In the second sub question, they told about Studman explained about gene mapping while working with Morgan. He used a frequency of recombination between the gene pair of the same chromosome as a measure of distance between the gene and map the position of the chromosome. This is more helpful to understand the amount of recombination and parental combination inheritance. For this narration, you will be getting another 1 mark. In this way, we can satisfy 5 mark demand of this particular question. In this 5 mark subjective question, they are asking us to explain about central dogma, in which way it was explained by Francis Crick, and also the exception with the reason and example. Here, first let me start discussing about the central dogma. The DNA multiplication into multiple number is called replication. Formation of RNA from DNA is called transcription. And formation of protein from RNA is called translation. For this mention, you will be getting 1 mark. For mentioning the yes, in some virus, there is a reverse transfer of genetic information because of the reverse transcriptase enzyme does reverse transcription process. In this process, the DNA is formed from RNA. For this mention, you will be getting another 1 mark. With the example HIV virus or retrovirus, you will be getting 1.5 mark. And for mentioning in which way the biochemical characteristic features of the transforming principle was not explained in Frederick Griffith experiment, that we need to explain in detail. Here, protein, DNA, RNA are purified from heat-killed S strain or smooth streptococcus or diplococcus pneumonia. For this narration, you will be getting half mark. Then we are adding protein and proteases to this mixture, so that what will happen is the transformation will occur. That means the R strain has become S strain. For this, you will be getting half mark. If you are adding RNA and RNase to this mixture, the transformation will happen. The R cell will become S strain. For this narration, you will be getting half mark. If you are adding DNA and DNase enzyme to this mixture, transformation is inhibited. For this narration, you will be getting half mark. Hence, the DNA alone is the transforming material. Like this, we can conclude. So that finally we can satisfy the remaining half mark to get 5 mark of this particular question.